So in this video lecture, we will study about the DNA sequencing methods. So there are different kind of DNA sequencing methods, and we'll discuss all the DNA sequencing methods in detail. So basically, what is DNA sequencing? The terms DNA sequencing refers to the method of determining the order of nucleotide bases like adenine, guanine, cytosine, and thymine. So basically, it is used to determine the sequence of nucleotides like adenine, guanine, cytosine, and thymine. So there are two kinds of bases in DNA. First one is purine, that is adenine and guanine, and second one is pyrimidine, that is thymine and cytosine. So I remember it like that, like all gods are pure. That means adenine and guanine are purines, and thymine and cytosine are pyrimidines. Now, what is sequencing methods? To determine the order of nucleotides, bases adenine, guanine, cytosine, and thymine in a molecule of DNA, two methods were used. First one is Sanger chain termination sequencing method. Second one is Maxwell-Gilbert chemical sequencing method. These two are most popular conventional methods. Robotic and automatic sequences are based on these methods. Now, what are the advantages and disadvantages? Suppose we want to see about the disadvantages of Maxwell and Gilbert chemical degradation method. So because it requires lot of purified DNA you will need, and there are lot of intermediate purification steps are there. It requires extensive use of hazardous chemicals. It is difficult to scale up and cannot be used to analyze more than 500 base pair. So the limit of the Maxwell and Gilbert method is 500 base pair. Now we'll see about the Sanger method, Sanger sequencing method. So basically it is a very great method, and it is designed by the Sanger sequencing method. So basically it is a very great method, and it is designed by the Frederick Sanger. So Sanger method was designed by the Frederick Sanger, and Frederick Sanger has developed a sequencing method and received a Nobel Prize for it. And now also this method is frequently used in laboratories for sequencing small genes, bacterial clone genes. All lab uses Sanger method because energy is very expensive. Also, not routinely Sanger is used in lab. Sanger sequencing method is also called chain termination method and dideoxy sequencing method. What it is Sanger method of DNA sequencing? We'll see why it is called dideoxy method of chain termination. The chain termination method is more efficient and uses fewer toxic chemicals and lower amount of radioactivity than method of Maxwell and Gilbert. So that's why Sanger method is very good. The key principle of Sanger method was the use of dideoxy triphosphate. Dideoxy triphosphate is very important because of presence of dideoxy triphosphate, there is DNA chain termination, and which helps in the DNA sequencing. Now you can see different kind of structures of the DNA. So first one is ribose. It is having two OH groups, and second one is deoxyribose. It is having one OH and one H group, and third one is dideoxyribose, which is having one H and H. So this is how dideoxyribose does not contain any OH. It is having only H and H, which is required for the synthesis of DNA, right? Now, how the DNA synthesis takes place? DNA synthesis requires the availability of the 3' OH group. As we have seen in the figure, there is no 3' OH group in the dideoxyribose. So it cannot lead to the synthesis of DNA. Chain will terminate during the DNA synthesis. So you can see in the figure how this DNA synthesis requires availability of 3' OH and energy. So basically there is a one basically OH group attacks the phosphate, and there is formation of condensation reaction is there, and there is phosphodiester linkage formation, and this will help in the synthesis of the DNA. So 3' OH group is pretty much required during the DNA synthesis. So Sanger method exploit this process by adding dideoxynucleotide for chain termination. Now, what is DNA synthesis? The absence of 3' OH on the 3' carbon of the sugar blocks further addition of the nucleotides. So this is the basic principle. As you can see, dideoxyribose blocks elongation. So there is growing chain and there is one base, and this is dideoxyribose. So dideoxyribose blocks elongation because there is no presence of reactive group. So synthesis is basically blocked. Now, chain termination method of DNA sequencing, it involves following components. First one, you will require a primer, then you will require a DNA template. DNA template is a must because without DNA template, you cannot do sequencing. And DNTPs will be required. DNTPs are basically dinucleotide triphosphates, collection of ATGC. DDNTPs will be required because you need chain termination. It involves 4 steps basically. First one is denaturation, then primer attachment, extension of bases, termination, and page, polyacrylamide gel electrophoresis. The method, basically the method involves, before the DNA can be sequenced, it has to be denatured. You have to denature the DNA so that you will get the single strand using the heat. So next a primer is annealed. Primer is annealed with the template, your denatured strand. The primer is specifically constructed so that its 3' end is located next to the DNA sequence of interest. Either this primer or one of the nucleotides should be radioactive fluorescence level so that the final product can be detected on the gel. So final product needs to be detected on the gel. That's why. Once the primer is attached to the DNA solution, it is divided into 4 tubes. First one is G, then A, then T, and then C. So G tubes all 4 DNTPs, DDGTP, and DNA polymerase. A tube contains all 4 DNTPs, dideoxy ATP, DNA polymerase. T tube contains all 4 DNTPs, DDTTP, and DNA polymerase. And C tube all 4 DNTPs, DDCTP, and DNA polymerase. So as you can see in the figure, Sanger sequencing procedure, there is DNA template, polymerase, access DNTPs, and primers. And there is presence of DDGTP, DDCTP, DDATP, and DDTTP. Now, the mixture of the DNA is first heated so that the DNA strands separate at 96 degrees Celsius. Then the temperature gets lower so that the short length DNA sequences a primer can bind at template at near about 50 degrees Celsius. The temperature is raised to 60 to 65 degrees Celsius, enabling the DNA polymerase an enzyme to bind the short sections of the double-stranded DNA. The DNA polymerase to synthesize new DNA. DNA polymerase will continue adding nucleotide to the chain until it happens to encounter a dideoxynucleotide. Dideoxynucleotide instead of normal one, so there will be a chain termination after encountering the dideoxynucleotide. So as you can see in the figure, sequencing of DNA by Sanger method, there is single stranded DNA with unknown sequence. That one is in blue, and it requires it acts as a template. Then there is DNA polymerase, then there is DATP, DCTP, DTTP, and DGTP. And there is radioactive labeled primers. And there are 4 tubes having DDATP for chain termination for ATP, and DDCTP, DDTTP, and DDGTP. As you can see, and while running on the gel, we have 4 lanes. One is DDATP, DDCTP, DDTTP, and DDGTP. Longer fragments will be at the upper part and shorter fragments will be at lower part. We read the Sanger from down to upward, from in 5' to 3' direction, and deduce the sequence of the template. Just complementary strand of this read sequence will give the sequence of our original template. So this is how the template reading basically takes place. So we go from down of the gel to upward, like 5' to 3'. Suppose we encounter first DDATP at lower part, then we'll keep it at 5' end. Then again we encountered one DDTTP, then we'll keep in the second position, and likewise we'll go from 5 to 3'. And the template strand will be just complementary to this strand. So this is how we read the Sanger sequence in the gel. Now, what are the uses of the Sanger sequence? So, reason up to 900 base pair you can amplify in length sequence using this method. In Human Genome Project, also, Sanger sequence were used to determine the sequence of relatively small fragments of human DNA. Sanger sequence is still widely used for the sequencing of individual pieces of DNA, such as fragments used in DNA cloning. Like suppose we want to clone a gene inside a vector and we want to just see whether it is cloned inside a vector or not. We have already confirmed it with the restriction digestion, but in order to confirm it very specifically to show our result, to show the proof, we use Sanger sequencing to see what are the sequences we have cloned inside the vector. So advantage of basic method, improve diagnosis of disease, identify suspect. Disadvantages are basically whole genome cannot be sequenced because small portion like 900 base pair you can only sequence. Very slow, it is very slow and time consuming also. So basically the comparison between Sanger and Maximum Gilbert method. Sanger method is enzymatic method, Maximum Gilbert is chemical method. So Sanger method DNA requires Sanger method requires DNA synthesis. Maximum Gilbert method breaks DNA different nucleotides. So automation is there in Sanger method and automation is not available in Maximum Gilbert method. So basically in Sanger method you need a single stranded DNA, and in Maximum Gilbert method you will need a double stranded or single stranded DNA. You can work with both. So what are the application of DNA sequencing? So basically in forensics, to help the identify individual, because each individual has a different genetic sequence. So you can identify who is the criminal and who is the means who is not criminal, who is innocent person. And medicine can be used to detect the genes which are likely to cause various genetic disorders such as muscular dystrophy. Agriculture, the mapping and sequencing of genome of organism helps to make them useful for the crop and food plants. Now, Maxwell Gilbert sequence is a method of DNA sequencing developed by Alan Maxwell and Walter Gilbert in 1976 to 1977. The method is based on the nucleotide-specific partial chemical modification of DNA and subsequent cleavage of DNA backbone at adjacent site to modified nucleotides. A Maxwell Gilbert sequencing process summarization. First, you label the 5' end of the DNA, helicode DNA samples in 4 tubes, perform base modification reactions, then perform cleavage reaction, perform gel electrophoresis, perform audio radiography, and you can see the results. So, chemical modification can be done by radioactive labeling at 5' end with gamma p32 ATP, which is added by the kinase. Then purification of DNA fragment to be sequenced. Chemical treatment generates breaks in DNA and run on the gel. Now, chemical modification cleavage. Polynucleotide kinase adds radioactive label at 5' end of DNA using a gamma p32, as you can see in the figure. There is 5' to 3' DNA sequence. Then p32 is added at the 5' end. Then chemical modification and cleavage. Base modification using dimethyl sulfate, like purine, adenine, guanine. Only dimethyl sulfate will add G. Dimethyl sulfate plus formic acid will add G plus A. Cleavage of sugar phosphate backbone will be done using the piperidine. Now, base modification using hydrogen. Pyrimidine like cytosine and thymine. Hydrogen plus hydrogen will having the cytosine plus thymine, and hydrogen plus NaCl will give only cytosine. So, cleavage of sugar phosphate backbone using again will be done using piperidine. So, this is the workflow. So, first you obtain the single stranded DNA. Second, you will add the phosphate p32 at 5' end. Then cleavage at specific nucleotides, like cleavage will be done at G, then 8 plus T, then T plus C, then C, and differentially sized DNA strand can be selected. And now you run it in agrogel electrophoresis. Mixture of DNA fragments of the different sizes will be run, and this will run from the cathode to anode, and you will compare longer fragments and shorter fragments. So as you can see, there is p32 labeled DNA sequence. First one is p32 level, and A plus G is clipped sequentially. You can see A plus G is clipped. In second one only G is clipped. Third one is only C clipped, and fourth one is C plus T. So this is an example of the Maxim Gilbert method. First lane we are observing only A plus G cleavage. Second only G cleavage. Third is only C cleavage, and fourth is C plus T cleavage. And we'll start reading from the down of the gel, just like the Sanger sequence. So first we have encountered C, which is the smaller fragment cleavage, then T, then A, then G, then T, then A. So this is how you get your sequence. And last one is pyrosequencing. Pyrosequencing is based on the sequencing by synthesis principle, whereas complementary strand is synthesized in the presence of the polymerase enzyme. So this is how template strand is there. Then first pyrophosphate ion released from the addition of the complementary bases, and then ATP sulfuryl adenosine is used. Then luciferin will add ATP. After adding ATP, you will give oxyluciferin and light. You will get a signal. So it initially uses immersion PCR technique to construct the colonies required for sequencing and removes the complementary strand. Next, a single-stranded sequence primer hybridized to endope the strand primer binding region, and the 4 different DNTPs are then sequentially made to flow in and out of the wells over the colonies. When the correct DNTP is enzymatically incorporated into the strand, it causes release of the phosphate. In the presence of ATP sulfuryl, the adenosine, the pyrophosphate is converted into ATP. This ATP molecule is used for the luciferase catalyst conversion of luciferase to oxyfurin, which produces light, and that can be detected with a camera. The relative intensity of the light is proportional to the amount of bases added. Peak of twice the intensity, 2 identical bases have been added in the succession. Pyrosequencing developed by 454 Life Sciences was one of the early success of next generation sequencing. Indeed, 454 Life Sciences produced the first commercially available next generation sequencer. However, the method was eclipsed by other technologies, and in 2013, new owners like Roche announced the closure of 454 Life Science and discontinuation of the 454 pyrosequencing platform. Sorry, it was 454 Life Science. So thank you. That's all in this video. If you have any questions, please comment in the section, and please like and subscribe the channel for more updates. I'll be happy to answer any of the queries by you.